Hello, this is Christopher Y. Tunick, and today I'm going to be giving you some info and a brief overview and kind of what's going on when it comes to mitosis. Mitosis is basically a pretty important part of the cell cycle, and overall it's a pretty small part of the cell cycle if you look at it, but it is honestly the most important when it comes to the actual division of cells. And it's part of basically the M phase of the cell cycle. But first, when it comes to mitosis, we got to start with interphase because that's where everything starts off. So there are three parts of interphase. There's the G1 phase, the S phase, and the G2 phase. So starting with G1, it builds the cells build most of the proteins, carbs, and lipids that they're going to need, for basic function, and they're going to perform their primary functions. So they're just going to be growing and doing their normal tasks. So there's going to be no cell division actually happening at this stage, or no prep yet. But they're just getting all the materials that they'll need to basically grow to a full parent cell. And then in the S phase of interphase, DNA is going to be replicated within the cell in the nucleus. It's going to basically be making an exact copy of the DNA that's currently in that cell. And histone proteins are also going to be synthesized inside of that cell in order to prepare for cell division. Now, in the G2 phase, that's the final phase, or final part of interphase rather, protein synthesis is going to jump. It's going to rather increase, and the cell is going to basically make its final preparations for cell division. And G2 is usually the shortest part of the cell, or a part of the interphase section, compared to G1 and S phase. So now we're actually going to move on to the process of mitosis once the cell is actually prepared. Getting ready for cell division, copied all its DNA, made sure it was actually perfectly copied, no errors, and then we can actually move on to the process of mitosis. And when it comes to mitosis, we first want to start with prophase. That's the first part, and it's going to start with chromosome compaction. So basically, it's going to take those really long strands of chromatin, like the chromatin fibers, and it's going to coil them up, and it's going to make them into nice short and thick bundles. And these bundles are basically going to be found in the nucleus, of course, and there's going to be a pair of sister chromatids. There's a pair of sister chromatids which are connected to the centromere in the middle between those chromatids, so they almost look like, I don't know, if you had these but connected at the end, so almost like that. And that center here that I'm talking about, right where my two fingers would almost meet, that would be the centromere. Here's a pretty good example. Give me one sec. We got a pen and a pencil. Let's use this for example. Here are our chromosomes. They're being our chromatin. They're these long, big fibers. They're being compacted down, and now they're all bundled together in these bundles. And there's these two sister chromatids, and at the center, as I drop the pencil, this center section right here where my finger is, that's going to be your centromere right here in the middle. And also mitotic spindle fibers are going to extend, and they're basically going to extend from each polar end of the cell and kind of connect to the sister chromatids. So they almost look like long strands, kind of connecting from each polar end of the cell, almost grabbing onto the sister chromatids. This is usually found in prometaphase. So now moving on to metaphase, the chromatin fibers are finally finished compacting down into those nice, thick, and bundled together packs of chromatin fibers, and they get moved into the center line of the cell, the metaphase plate, right down the center. And they're also, while making sure they're all connected to the spindle fibers, and it basically prepares to separate the parent cell and separate the sister chromatids into two, basically daughter cells. And finally, next in anaphase, the sister chromatids are basically going to actually separate, and they're going to split in two. And they're going to be pulled to the opposite side by the spindle fibers to basically separate the two sets of chromosomes and have it so that each daughter cell has an exact set of those chromosomes for each. Then in telophase, which is the actual final phase of mitosis itself, the sister chromatids are going to be finally pulled to each end of the pole. They're done actually moving at that point. And they're going to kind of unwind slightly. They're going to lose that sort of compactness and tightness that they had as those bundles, and they're going to begin to unwind almost like how you would see in the nucleus, like those long strands together, they're going to begin to unwind. And the spindle fibers are also going to disappear because they're not needed anymore. Those are just going to kind of fade away. And a nuclear envelope is going to basically form around each set of the chromosomes. The final step of mitosis, or rather not mitosis, sorry, the cell cycle, is going to be cytokinesis. Now, this is a very important part of the cell cycle, as it's the actual division itself of the two cells. It usually overlaps with mitosis itself in telophase near the end, so it kind of just starts at the end of telophase. So they're kind of at the same time. So normally on like animal cells or like fungi, like a contraction ring is going to be formed around like the cell, and it's going to contract the cell together and almost like pinch off the cell into two separate daughter cells. Compared to like a plant cell, which is basically going to create like a cell plate out of like vacuoles, or rather, sorry, it's going to be a line of vesicles, which are going to like fuse to become like the cell walls of each cell. So they have different processes when it comes to cytokinesis. And this process is actually going to produce two daughter cells with with two sets of chromosomes, which are also known as diploid cells. And which I stated prior, they have the exact same amount of chromosomes, or rather the exact copies of the parent cell's chromosomes. And it's super important, at least mitosis is, in terms of just general function of a lot of organisms, when it comes to growth and repair, when it comes to like an actual, like a person or an organism like growing, it needs to create more cells in order for growth, so it needs to divide those cells and create more. And also for repair, if there's something damaged, it would need to basically duplicate the cells that were lost by cell replication, which is mitosis. So that's a general overview of mitosis. You can see how important it is when it comes to life processes, and that's my general understanding of it. Thanks for listening. Have a great day. Thank you.